Hi everyone, thank you for spending your time on our unethical airflow training. I'm Huimin from U School of Medicine Clinicians Lab. Today I'd like to introduce you our unethical airflow pipeline. I will introduce what is AIR and what is AIRR-seq and our pipeline, and we will have some hands-on practice and to interpret our results, and also I will answer some frequent asked questions. But for the sake of time, let's first create GitHub code spaces and run test profile. GitHub code spaces is a cloud platform with GitHub. There it has all the packages and the container pre-installed for you, so you can just run to test our pipeline. So let's first go to our tutorial. I already have it open. So the tutorial, you follow the tutorial, you can learn how to run an unethical airflow pipeline. First, if you want to create a code space instance, you need to go to a GitHub repository that you want the instance to be based on. So here, let's go to our unethical airflow repository. Here you can see here is a code button. You click it, and it will show local or code spaces. So you can click plus, and it will create a code space. But by default, it gives you only 2 CPUs. So we want more, so we need it with more options. So click new with options. And here, yeah, we want the platform on unethical airflow master branch, and we want it with 4 core. So by default, it only has 2 core, 8 GB RAM and 32 GB storage, not enough for our test. So we want 4 core. Create code space. This step usually takes 2 or 3 minutes. So let's have a look of what. And also, I noticed that someone may not have the code space here. That's because you never use it on your GitHub. So so you need to make some agreement on GitHub to use it. So you can go to GitHub code space page, and it will pop up some agreement, and you click agree, and then you can just from here click new code space. So it's similar to go to a repository and click create new code space, but here you need to type in the repository where you want to start your code space. Let's have a look. Let's see whether our code space created. No, it still needs some time. So let's go to my GitHub. Okay, in your GitHub, there will be, you click here, there will be a tab, say code spaces. You click here, you will go to the GitHub code space page. So you can, if it's the first time you use it, you need to agree something, and then you can create new code space from here. Okay, let's see whether mine is built. Okay, after that, if the environment is ready, you can type in, you can check whether you have all the packages needed for unethical airflow pipeline. Let's see what to check. Here we have already opened it. And then to verify you have installed the Singularity, Nextflow, and all required dependencies, you can run Nextflow with test profile in your code space terminal. Okay, it's ready. So let's test. But if you want to see whether you really have Nextflow pre-installed, you can just type Nextflow -v, that is to check the version. Okay, it says I have the Nextflow version, which means my Nextflow is installed here. Yeah, and then as I said, you want to check something else, like whether you can run airflow pipeline. But first, probably you can set up a singularity cache directory. In that case, all your airflow run will be, the singularity will be stored here. So you do not need to pull the singularity every time when you run the pipeline. So make directory and export it to singularity directory. Okay, let's do it. So it's here, yeah. Just allow. Okay, now you see there is a new directory created, means has a name singularity cache. Okay, let's run the test. Nextflow I call airfly. Here I used the dev version that is in our development branch. But when you really run our core airflow pipeline, please check our latest release. Currently, we have 4.3.1, but we will have a new release 4.4.0 very soon. So make sure to check the latest version. Control-C, and let's go to our, where is here? Okay, it's right. Now you can see it shows Nextflow version and pulling something, so it's running. Okay, now let's go to our introduction of what is AIRR-seq and what are we doing now. Where is my slides? Okay, here. Okay, let me. Okay, we have already tried this code. Now I want to introduce AIR and AIRR-seq to you. What is AIR? AIR is adaptive immune receptor repertoire. It is a collection of B cell receptors and T cell receptors in an individual. BCRs in their secreted form are also called antibodies or immunoglobulins. Their role is to bind antigens and enable an adaptive immune response. There is estimation of 10 to 11 unique receptors at a given time in the human body. The high diversity of receptors allow binding to a wide variety of antigens. BCRs are composed of 2 identical heavy chains and 2 identical light chains. The heavy and light chains are generated from VDJ recombined genes. That means the germline genes actually is not like of a single V gene, D gene, and J gene. There are a lot of repeats of these segments. On the germline, your BCR heavy chain region has a lot of variable gene segments and diversity gene segments and then joining gene segments. And during the VDJ recombination process, and random, 1 V gene and 1 D gene and 1 J gene will be combined together to form the variable region of the BCR heavy chain. The light chain is the same, but the light chain does not have the D gene. So this region can also be divided in another way. There is a framework region or the CDR region. CDR region is complementarity determined regions. These regions are directly bound with antigens. So, and also BCR has constant region. Constant region comes in, it determines the function of the BCR of different constant region. We see the BCR of different other type. So here is TCR. TCR are composed of an alpha chain and a beta chain, or in some rare cases, a gamma chain and a delta chain. Each chain is also generated from VDJ recombined genes. It also has framework region and the CDR region that are most important for binding the antigen, especially the CDR3. BCRs can directly bind antigens, but TCR bind antigen epitopes that are presented on MHC by antigen-presenting cells. I will not go to the details. And a very important analysis in our AIR analysis is to identify the clones and the distribution of the clones. So what is clone? Clone is a group of sequences that have the same ancestor. So to understand this, we need to know the development of B cells and the B cell expansion. So as we said before, after VDJ recombination, we will get our naive repertoire. These B cells. They have diversity, but many come from the recombination of VDJ gene and random. And they are waiting for their cognate antigen to bind with them. And once one antigen bind with the BCR, it will start the clone expansion. And at the same time, there will be somatic hypermutation. That is a mutate of the nucleotide that to increase affinity of these BCRs with the antigen. So we will get a lot of B cells that are specific for this antigen. But these B cells, the sequence of this B cell, the sequence of the BCRs are not identical due to the somatic hypermutation. Meanwhile, the B cell will undergo class switch recombination. That is, change the C gene, change the other type. And cell subtype differentiation. That is, some B cell will go to memory B cell, and some B cell will differentiate into plasma cell. So that's why we want to, it's not easy to find the B cell, the BCRs from the same clone because the sequences are not identical. But for T cells, after antigen stimulation, there are clone expansion, but there's no somatic hypermutation. So for T cells from a specific clone, the sequences are identical. So why do we need to, why do we want to study AIRR-seq? Because the AIR can potentially reveal immune signatures associated with past infections, current infections, and ongoing immune-mediated disease. And the sequencing, the AIR has a multitude of applications, like studying the immune state of an individual, to study the developing immune diagnostics, to develop the immune diagnostics and to guide vaccine development and to develop immunotherapies. And to make this feasible, we need to appreciate the sequencing technology. Now, like RNA-seq, for AIRR-seq, we also have bulk and a single-cell AIRR-seq. For bulk AIRR-seq, after we have the messenger RNA, we will use V primers, C primers, or V primers, J primers to only get the BCR or TCR messenger RNA. And then we will sequence these to get the bulk AIRR-seq. It is high throughput. However, there's no paired heavy chain and light chain. And also we do not have cell information. And we also have single-cell AIR sequencing. Single-cell AIR sequencing, it's the mostly used technology is a 10x. Probably you are already familiar with single-cell RNA-seq. Like for here is a 10x barcoded gel bits, and it will in this device it will be combined with one or zero or probably two cells, and then there will be an oil droplet rapid. So in most cases, it's just one barcode and one cell. So all the sequence in this droplet has the same cell barcode. This is how to differentiate messenger RNAs from different cells. And this is how to prepare for the single-cell RNA-seq. But to get the AIR-seq, we also use V primers and C primers or J primers to only amplify the BCR or TCR sequence. So the pros of the single-cell AIR-seq is that it has paired heavy chain and light chain, and also it has the cell type information from single-cell RNA-seq. But it compared with bulk AIR-seq, it is of low throughput. If you are familiar with RNA-seq, you know, like the first step is we map the reads to the reference genome. But for AIR-seq, it's challenging because, as we said, on the germline gene, it's not of a single V gene, a single D gene, and a single J gene. It has very highly repetitive V genes, D genes, and J genes. And also, there are high allele variability across individuals. So we need specific tools to do the AIR-seq analysis. That's what we do in our lab. In Kleinstein's lab, we have developed and we are maintaining a lot of tools. It's Immcantation framework that we have a bunch of toolsets to do AIR-seq analysis. Like Presto is to do sequence assembly. And Change-O is VDJ alignment with the IgBlast and IMGT, and Scoper to do the clone analysis, and Dowser, the lineage tree analysis. So if you want to do AIR-seq analysis, one choice is that you go to the different tools to learn them one by one, but it will cost you a lot of time. That's why we have our NF called airflow pipeline. There is you can do the analysis from start to finish with just one line command. So here is the structure of our workflow. You can do it with single-cell or bulk AIR-seq, and from the raw data, the FASTQ files, or if you have the assembled data, you can start from the assembled data. We mainly do the QC and sequence assembly, VDJ assignment and filtering, clone analysis, and germline, lineage tree construction, and some reports. So IF core airflow is written with next flow. Next flow is a system, therefore pipeline. That it is portable because it supports a variety of computing infrastructures. You can run it on your own laptop or on HPC or on cloud, and it is reproducible, and it support multiple container engines like Docker or Singularity or others. It has continuous checkpoints when you run the pipeline. So if the pipeline fails at a point, after you fix a bug, it will not go back to the beginning. It will start from the failure point. And also a lot of process are in parallel. And also, IF-core airflow pipeline belongs to the IF-core community. It follows the rules of the community. So we have very stable releases, and we have extensive documentation. We have continuous integration tests and community code review. Here is another map of our IF-core airflow pipeline. So you can see the detailed process here. So I will not explain them one by one. It will cost me a whole day. Okay, and the requirements to run IF-core airflow pipeline, you need to have Linux system or Mac. It does not support Windows now. And you need to download the Nextflow, and you need the container. Now, mostly used is Docker and Singularity. So okay, that's the introduction of AIRR-seq and IF-core airflow. Let's see whether our test profile runs successfully. It's still running. Okay, I think probably I can kill it. Of course, I know it will be running successfully. So after kill it, let's run a real test. This is we run it with a single cell data from the assembled data. So if you want to run the IF-core airflow pipeline, you just need one line command, probably a long line. So next flow run IF-core airflow -r, that is the release. Here we use def, but you can use the latest 4.3.1 or in the future 4.4.0, and profile on Singularity on code space. It only supports Singularity, so we use Singularity. But most probably on your own laptop, you will use Docker, and we recommend you use Docker. In the mode, we say this is assembled data, so assembled. And you also need a sample sheet. The sample sheet it has the required columns, and you can check our IF-core airflow website to see the requirement of the columns of the sample sheet. So like the file name, the species, the subject ID, sample ID, tissue, 6H, by provider, target locus, is it IG or TR, single cell, that is whether it's single cell true, as we have single cell data here, so it's true. If it's bulk, you put false here. And specify the where to store the results. We want it to be in the folder named results, and the configuration file. It's optional. If you want to have better control of your pipeline, probably, or if you want to specify the resource you need the configuration file. Here we only, because in code space, we only have 4 CPU and 16 gigabytes, and the usable is less than 16. So we set memory here 15 and 24 hour and 4 CPUs. And in code-space terminal, you need to go to the directory where the example data provided. Yeah, we provided the example data, but you need to go to this page, go to this directory. Let's go there and run this pipeline. Okay, let me see, directory. Okay, it's here. Now you can see we have all the files that we need to run the pipeline. Let's go to check. Okay, here it requires 16 gigabytes and 80 CPU. It's more than the platform can provide. So let's change it. 15 and 4 CPU. Yeah, I use the command to change it, but if you are not familiar with it, you can just go to the files, open it, and change it and save it. Okay, and also there's one thing that I need to have a look, because we will also provide the bash file that you need to just run the pipeline with it. So here, let's double check. Okay, now here it uses version 4.2.0. Let's try our latest version 4.3.1, or you can use Dave. There is a development version, profile. It only supports singularity in code space, assemble data, configuration. Okay. Save it. Okay, we can run it. Okay, it started. So how will the result look like? You can see after it started, I think now there will all. It's in here, docs, usage, single cell tutorial. We write from here, so the results is here. Here you can see the results. After running, there will be a bunch of directors and results created. And of course, you can just view the results here, but we recommend you to download the results. Just click down, right-click it and download. The reason we want you to download is first, we have a lot of HTML files like for our report. It does not look very well on code space. And secondly, after you close the code space, you will lose all the files. So we recommend you to download it. So I already downloaded it, so you can have a look. This is results from the test profile, and this is the results for the assembled results. You can see we have the airflow report, and also we have a lot of the results for other steps. So how to interpret this result? Let's have a look. Yeah, this is how to run it. After run it, don't forget to delete it. You can go to the code space, GitHub code space page, and below there it will show all the code space instance you opened. You just right click here and delete. Otherwise, you may receive the bill because it's not for free for whatever amount of tasks you want to run. It has a limit on the running time and the resources. Okay, let's move on to the results and interpretation. As I show you, there will be a file named airflow report HTML. It is a summary of the report. In it, you can see like the number of sequence per sample after each of the steps. So from here, you can get idea like at which step the sample lose a lot of sequence. So you can go back to check the reason. And also we have like, for example, V gene family usage for each sample. And we have a lot of subfolders. It contains the intermediate results. For example, the VDG annotation, it contains all the results of the VDJ annotation. The first one is after running igblast, it has the results, and the second one is to convert the igblast result to our the table that required for the following analysis. Also, like some of the filtering and like select productive, keep only productive sequences. And another folder has named QC filtering. In this step, for single cell, cells with heavy chains or with multiple heavy chains are removed. For bulk, it will do collapse duplicates of the sequence and remove duplicated sequence. And then a very important folder that you need to check is a clonal analysis. Within it, there are 5 threshold clonal assignment. 5 threshold is to estimate the optimal distance threshold for identifying clonally related sequences. So this is like when we do clone assignment, we want to group sequence below a threshold together to consider them as they are from the same, they are related, they are probably from the same clone. And the sequence above the threshold as because they are far away from each other, so they are not from a same clone. So how to determine the threshold? So for each subject, for each sequence, we will calculate the distance of this sequence to its nearest neighbor. The neighbor is the one that is mostly like this sequence and also have the same V gene, J gene, and junction length. And after we get the distance matrix, we will do this plot of the distance of all the sequences with its nearest neighbor. Above x-axis is within one subject. So usually it is bimodal. The first mode are sequences that can find a sequence within a same clone. So they can find a sequence that it has a small distance with it. And the second mode are most likely sequence from singleton. Singleton are clone of only one sequence. It's before clone expansion. So we want to find a line to separate these two modes. And below y-axis is the distance across subjects. It will give us a guide, like whether our threshold makes sense, because most of the time we do not want sequence from different subject below this threshold. So like here, the estimate threshold is 0.23, 0.28. We feel it's a little bit high. And you can see there are sequences from different subjects below this threshold. So typically, our experience tell us usually the threshold is around 0.1. So here, probably I will recommend to rerun the pipeline with threshold 0.15. It makes more sense. Yeah, so we need to calculate the threshold. By default in unethical airflow, it's calculated automatically. But if there is a failure to calculate the threshold due to like the not enough sequence, you can set it manually. Or if you, like I said, if you are not satisfied with the estimated one, you can set it manually to rerun the pipeline. And also the final threshold, because we calculate threshold by subject, the final threshold is we used for clone analysis is the mean. So here it's the mean of 0.23 and 0.28. But this is for BCRs. For TCRs, we said that from the same clone, the sequences are identical. So you just need to manually set the threshold to be 0. Okay, after we get the threshold, we will do clone assignment. So in this step, we use hierarchical clustering. First, we have all the sequence from a subject, one subject together. That is from one donor, and because like most of the time we want to, like before vaccination, after vaccination, we want to compare the clones, the change of the clone. So we want the clone ID to be unified, like in a single subject, a single donor. So we group all the sequences belong to this subject together, and then we have VDJ partition. That is, the sequence have the same V gene, J gene, and junctioners will be further grouped together, and then we will do the hierarchical clustering, and then use the threshold we calculated before to separate the sequences. So to cut the tree structure to get clones. Yeah, this is how we do clone analysis. After clone assignment, we have the clones of each sample. So we will do some analysis on the features of the repertoire. Like here, like we have the clone size distribution, number of clones, and also we have clone abundance. Clone abundance is different from clone size, clone distribution, because in that one, we only have the clones that we detected in our experiment. But clone abundance, it not only has detected clones, it will also infer the unseen clones. Like here, this is our assembled data. It has two subjects, S1 and S2, and it has two time point, D0 and D12. D0 is before vaccination. D12 is 12 days after vaccination. So you can see that the clone abundance increase after vaccination, which means after vaccine, some clone were expanded. And in clone assignment report, you can also see plots like the mutation frequency. That is the, like in each sample, the mutation frequency of different C gene of different either type. And in repertoire comparison folder, you can compare several repertoire characteristics such as V gene usage across subjects, time points, or cell population. Here is, we can see the V gene family usage across different samples. Yeah, this is the results interpretation, and now I want to answer a most asked questions. There is what can be said in the custom configuration file. Like here, you can set the resource you want for a specific process. Like here in change of assigns, I want to CPU 10 gigabytes and 5 hour. Or for example, the gene assignment usually cost a lot of memory and CPU. Probably you figure out you need 800 gigabytes. So you can require that amount there, but you need to make sure you have your you have that resources in your system. And also you need to set the resource limits, because resource limits is a cap of all the resources you can ask in your process. If you want 800 memory in one process, but here the memory is 72 gigabytes, you can get at most 72. So don't forget to change the resource limits to 800 gigabytes memory as well. And also, like it's for this pipeline, if you run on large data set, a lot of samples, it's very common to have error. So it's probably like one sample with low quality. If it has an error, the whole pipeline will fail. So if you want to prevent it from happening, you can set the error strategy to be ignored. So it will ignore the error from this process, or you can ignore the errors in the whole pipeline running. Yeah, here you can see a list of other error options in the Nextflow documentation. Yeah, that's it. If you have any questions, you can go to our NF-core airflow documentation. We have a lot of things that you can check and you can learn from it. And also you can go to our GitHub page. Also there is a NF-core Slack. There's a channel called Airflow. You can join it and leave message in our channel. Yeah, here are the materials that I mentioned, like the airflow pipeline documentation and NF-core troubleshooting. And here is our Immcantation tools. So thank you for attending the training. And if you have any questions or feedback, you can contact me. Here is my email address. Okay, thank you so much.